Thank you, Chairman, for the introduction. Thank you to the organizers for giving us the opportunity to present our work. So today I would like to discuss with you a novel methodology called functional respiratory imaging that we've been developing over the last couple of years to look at the bioequivalence of inhaled medication. So first I would like to demonstrate the technology or discuss the technology a little bit, also the added value in the drug and device development, and then show a bioequivalence case that we did the last couple of months. So in the respiratory space, we actually have quite a number of challenges. So one of the challenges is that the limited sensitivity of the current outcome parameters, in particular the FEV1, the forced expiratory volume in 1 second, actually leads to very difficult registration procedures for innovators on one hand, but also very difficult procedures to demonstrate bioequivalence. So that forced expiratory volume in 1 second is basically the amount of air you can exhale in 1 second, and that's the gold standard today in the respiratory space. So you can imagine it's quite a crude method. It's quite a noisy test. So what we believe is that by using imaging, and more particular functional respiratory imaging, we can actually have more sensitive endpoints. So we have enhanced sensitivity compared to the pulmonary function test. And of course, if you have more sensitive endpoints, we can reduce the number of patients in those clinical trials. On one hand, to maybe de-risk the very expensive phase 3 trials for innovator products, but also to assess bioequivalence, and that will be the topic of today's talk. So what is functional respiratory imaging? So we start actually from the high-resolution CT scans that are available in most modern centers nowadays. Then we do the segmentation, so we actually segment and make three-dimensional geometries from the anatomical structure of interest, and I'll show you some of the examples later. And then we can combine that with simulation of the flow, of the inhaled flow, a method from the aerospace engineering field, where we can actually derive resistances, but also aerosol deposition characteristics. So as mentioned, we start from the high-resolution CT scans, and we combine that with the so-called computational fluid dynamics, which is basically solving mathematical equations, and that gives us flow properties throughout the entire domain, flow properties like the velocities, but also the resistances, the velocities, the pressures, the densities, and that allows us to calculate the resistances. So what are these endpoints? So if we start from 2 CT scans, one at expiration and one at inspiration, we can actually have a whole range of outcome parameters relating to the ventilation, relating to the perfusion of the tissue, and relating to the deposition. So for instance, some of the parameters that relate to ventilation are the lobar volume. So here you see the lobes at expiration, so functional respiratory capacity, so it's lung level after a normal expiration. And this is basically the lung at total lung capacity. So we can look at the individual lobe volumes, and we can really see whether these individual lobe volumes are the correct size or whether they're hyperinflated or hypoinflated. We can also look at air trapping, which is a characteristic in specifically smokers' lungs, COPD, where we can actually visualize and quantify the regional expression of the air trapping itself. We can also look at the airway volume. So here, for instance, you see the airways down to the level of the smaller airways at total lung capacity. So we can look at individual branches, see airway narrowing, for instance. You can also look at the expiratory scan, and then you see things like airway closure, air trapping, etc. So quite detailed approach, a quite detailed description of the respiratory system. We can use the computational fluid dynamics, so the flow simulation, to look at the regional resistances. So we have the resistances of the larger airways as well as the characteristics of the smaller airways. So for instance, here you can see the changes after bronchodilation. So if we take a CT scan at baseline, if we then administer, for instance, a bronchodilator and we take a new CT scan, we can actually extract the 3D geometries at the baseline and at the follow-up scan. We can then overlay those two geometries and look at the regional differences. So for instance, here this is an asthmatic patient after bronchodilation, and you can see where the bronchodilation actually occurs. For instance, here you can start comparing branded products versus generics, as we will see later. So very highly detailed description of what happens in that system. For the same patient, you can look at the changes in the resistances here. So the color codes here indicate a reduction in the resistance after bronchodilation, of course. So you can start comparing the different parameters. We can not only look at the ventilation itself, we can also look at the perfusion and at the tissue. For instance, we can look at emphysema. So emphysema is a destruction of the lung tissue, again, often seen in smokers' lungs. And we've developed an algorithm that basically filters out the normal air from the affected tissue. So if you apply the algorithm to a normal volunteer, you don't see any of the black dots. But in a COPD patient, so in a smoking patient, you actually see the regional presentation of the emphysema, of the destruction of the tissue, on a lobar level again. So again, very detailed information. We can also do the same for the blood vessels. So here you see the segmentation of the blood vessels to assess the blood vessel density in the different patients. And similarly to doing the bronchodilation, you can also look at the vasodilation. So here you see the changes in the blood vessel volumes after the administration of a vasodilator, after the administration of nitric oxide. So again, you can look at the regional changes. We can also look at airway wall thickness to see whether there is inflammation. So here you see the 3D reconstruction of the lumen itself, where the air passes, and then the light gray is actually the airway wall thickness. We can also look at depositions. So without the need for labeling or for using gamma scintigraphy or radiation, we can actually couple the individual geometries that are derived from the CT scans with any type of inhaler. So for instance, here we coupled the same patient geometry with a DPI and then with an MDI. And then we can use the computational fluid dynamics, so the flow simulation, to look at the regional deposition characteristics of the different products. So here we switched from larger particles to smaller particles. Then you can also see that the deposition concentration reduces when you switch to these finer particles. We get more particles in the lower airways. If you know the upper airway deposition, you can also look at the effective lung dose, so how much of the particles actually reach the lower airways. And you can imagine if you want to compare generics versus branded products in the inhalation space, that's quite a crucial parameter. So why do we want to do this? Typically you have here, it's a little bit difficult to see, but you have the drug at the site of action. And that drug typically has an effect on the geometry. You want to open up the airways to make the patient breathe better. So it should have an effect on the pulmonary function, easier to breathe, and that should have an effect on the quality of life. Now, if you look at the different categories, you see that the more downstream you go, the higher the number of confounding factors are. The lung function typically also is determined by the muscle force, the patient effort, etc. You have the comorbidities that influence the quality of life. So the higher the number of confounding factors, the larger the sample size needs to be in those clinical trials. And that's often why the clinical trials in the respiratory space are so expensive. So in order to develop, let's say, more cheaper generic drugs, it's quite beneficial to be able to reduce that sample size using more sensitive endpoints. You see the additional sensitivity if you go closer to the site of action in this graph. So what you see here is basically the change in the forced expiratory volume in 1 second, so the change in the gold standard parameter, versus the change in the CT-based parameters, so the high-resolution parameters. And you see overall there's a significant correlation between the two. But if you look at the slope, you basically see there's a factor 3. So that means if you use the conventional endpoints and you see a 10% difference, that results in about a 30% difference in those image-based parameters. So you actually increase your signal strength. You do that in terms of the volumes, and you do that also very much in terms of the resistances with the flow simulation. So there the factor goes up to almost a factor 4. So you really increase the signal strength of what you're doing in the respiratory space. Of course, if you improve the signal-to-noise ratio, that has an effect on the sample size. So you can, for the same power, you can reduce the sample size from about 70 patients if you're using the classical endpoints to about 25 if you're using the FRI, so if you're using the imaging points. This was in asthma study. And in a COPD study, the sample size calculation indicated you could reduce from about 100 patients to about 16. So you can see a lot in smaller clinical trials. So how can we use that for bioequivalence? So recently we did a clinical study where we looked at the generic versus the branded salmeterol fluticasone, so an ICS-LABA combination, so a combination of an anti-inflammatory drug with a bronchodilator. Because this is the hypothesis basically. So here you have the conventional endpoints, the FEV1, so the PD endpoint, and here you have the pharmacokinetics. And we believe that the imaging tool can actually bridge between the PK and the PD, because typically the PD has a low ability to differentiate products because it's a relative insensitive endpoint. But in the PK has quite a high ability to differentiate products, but it's often too sensitive, so it's often difficult to demonstrate the bioequivalence. So by providing more information from the PD to the PK, I think we can facilitate the development of these products. So what did we do? We did a double-blind, double-dummy, randomized two-way crossover study with Seretide, which is the test product, and the reference product is then the branded product, basically. We did this in 16 asthmatic patients, and we used FRI as an endpoint. So I really wanted to see whether the test and the reference products are the same in terms of these image-based parameters, including the CFD parameters. And we also looked, of course, at the other parameters such as lung function, exercise capacity, dyspnea, and also some safety. So this was the study design. So at the first visit, pre-bronchodilation, we looked at imaging, the conventional lung function test, and exercise tolerance. And then 2 hours after the administration of either the test or the reference, we repeated everything. And then we also repeated the same procedure after 1 week, of course, switching the test and the reference, really the crossover design. We looked at the CT scans. We also measured the inhalation profile to see whether patients actually had a different inhalation profile with the branded versus the generic. And we also looked at a set of the pulmonary function tests. We wanted to assess whether the products are different in terms of the Wilcoxon matched pairs to assess the drug effect, and also the Mann-Whitney test to compare the drug effect. So we wanted to see from the pre to the post, is there a difference? Is there bronchodilation effectively? And also to see whether the generic and the branded product were different in terms of their bronchodilation. We also wanted to see whether the products are equivalent. And to do that, we did a 90% confidence interval and to assess whether they're in between the 80 to 125% bounds. So these were the inclusion criteria, typical asthma patients. So the randomization scheme, a list of the patients, and if we look at the results, so typically we have for all the patients in the study this type of images. So we can look at the deposition of the test product and the reference product. We can look at the actual bronchodilation, so this is the change in the volumes from the test, the reference, and also the baseline stability, so visit 1 pre versus visit 2 pre. And we can also do that for the resistances with these sensitive endpoints. So here a little bit larger. So here is the same patient with the test and the reference product. So you can actually start comparing whether there are differences in those regional characteristics in terms of the volume as well as in terms of the resistances. So if you look at the overall parameters, so here you basically see the inhalation maneuver. You don't see any difference between the test and the reference product in terms of the inhalation maneuver. So typically the patients use the 2 inhalers very similarly. They were both MDIs, so that's not so surprising. Also in terms of the drug deposition, there was no significant difference between the 2 products. So you can see also here from the aerosol deposition characteristics. So if you look at, so overall you could see that the drugs do very similar things. The question is also, they're not significantly different, but are they equivalent? And in order to do that, we wanted to see whether the technology could actually pick up the difference between products that are not the same initially. So we did another clinical study where we compared salbutamol with ipratropium bromide in a small cohort of COPD patients. And the interesting part is that if you look at the classical endpoints, it's a little bit difficult to see, but the classical endpoints, on average, they don't show any difference in terms of their FEV1. Also, in terms of their volumes and resistances, they're pretty similar. But if you look at an individual level, you actually see that some patients have a very good response to one product and a limited response to another, and vice versa to the ipratropium bromide as well. So if you look at the overall characteristics, so this is the Atrovent versus Ventolin, so 2 products that are not the same. This is the 80 to 125% confidence interval. You see that the FEV1 is not able to distinguish the 2 products, but if you're using the FRI-based endpoints, you have the increased sensitivity to differentiate between the 2 products. So these are products that are not the same. So if we apply this to the current clinical trial, you actually see that those bars reduce quite substantially. So you're starting to see the differentiation again in terms of the sensitivity, but now they're much more the same. So if you compare them 1-to-1, so these are the two products that are not the same. So you see the ability to differentiate between the two products, and if they're the same, also the technology will tell you that. You can also add the deposition to that to see whether the deposition between the branded and the generic is similar or not. So in conclusion, I think we can say that the functional respiratory imaging technology can actually facilitate the bridging between the PD and the PK. So I think it opens up a lot of possibility to develop maybe better generics, improve the introduction of generics into the market using these smaller clinical trials. So in conclusion, we can say that FRI is more sensitive compared to the standard PFTs. We can have that regional information that really increases the signal-to-noise ratio, and we can potentially bridge between the PK and the PD. So thank you very much for your attention, and if you have any questions, please feel free. Thank you.
What's the current FDA criteria for bioequivalence between the inhalation?
So the FDA actually basically requires quite a lot of things. You have to show in vitro equivalence, you have to show PK equivalence, and you have to show PD equivalence. So for the PK, you can do a 2-way crossover with 1 dose, but for the PD, you actually have to do a 4-way crossover. So you have to do 2 doses of the reference and 2 doses of the test product. So it's quite an extensive study protocol that you have to do. So the idea is here, if we have more sensitive endpoints, we could potentially also make those clinical study design protocols simpler so that you can have a quicker development trajectory, basically.
So FDA accept the FI-based data?
We're in the process of talking to them to use this as a development tool, yes, as a biomarker, basically.
Have you developed the software to this 3D-dimensional imaging, or is something common that is coming with the equipment?
So the FRI workflow itself, that's something that we've developed in-house. So typically the way it works, so nowadays with the data centralization, we collect the CT scans from everywhere in the world in a central location, and we provide, we do the workflow, and then we provide the results according to standard operating procedures. So it's not a software that is sold or delivered. It's more of a, let's say, a lab approach where you send a blood sample to a lab and you get the results back. Here you send a CT scan to a central lab and you get the results back. And that ensures a reduction again in the variability of the results, of the processing itself, and it increases basically the value and the accuracy of the results.
Can you give us some basic principle of the software you're using?
Yeah, in terms of the principles, so you have 2 aspects. You have the segmentation, which is basically, it builds up the voxels from the CT scan, so it gives you the anatomical information. And then you have the flow simulation. That's basically solving mathematical equations, and that gives you resistances and aerosol deposition characteristics, if that is what you refer to as the principles.
So the current study actually you utilize the study to validate your FI methods.
Yeah, so this is a study that looks at the bioequivalence, but we've done quite a lot of studies in the innovator space, really looking at the effect of novel products and also in a crossover design. So for instance, novel product versus a standard of care, and see whether there are differences in terms of those endpoints. So here it's more the equivalence approach.
So here is, so have you validated your FI as the effective marker for?
Yeah, absolutely. So we validate with, for instance, a SPECT CT scan in terms of the deposition. We've also compared it, of course, with the pulmonary function test, with the body plethysmography, also with the gamma scintigraphy and the hyperpolarized helium. So quite a comprehensive set of validation studies that we've done, and we also published that. We've been doing this for about 8 years now, so quite a long time. Yes, we're neighbors, actually.
So, one additional question. Have you applied this surrogate endpoints as efficacy endpoint for some agencies, maybe EMA or FDA? Have you used it?
Yeah, so now the first files will be submitted with this evidence or with these endpoints as supportive evidence. So we've been talking to the FDA, and they say, if you do it in a phase 2 design, we definitely accept this as supporting evidence. So we will look at it, we will assess both the efficacy based on these novel endpoints and the gold standard endpoints. And we're moving forward to really biomarker qualification so that it can also be used as primary endpoints. But the first files there will be submitted very shortly with this technology as supporting evidence.
I congratulate Jan D. Becker for giving his presentation as well. Thank you.
Thank you.